Janet. All right, so our, it just covered my whole screen. Okay, sorry. So tonight, our speaker is Janet, and Janet is the owner of Bat Survey Solutions, which is in Tucson, Arizona. She got her start working with bats while studying the pollination biology of columnar cacti in Mexico. She has worked for Merlin D. Tuttle at the Bat Conservatory International for over 15 years, teaching bat survey field technique workshops and educating others about bats, including annual training at the Southwestern Research Station in Portal since 1992. She is the author of The Bat, or the Bat chapter actually, in books Cave Creek Canyon, Revealing the Heart of Arizona's Chiricahuan Mountains, and A Natural History of the Sonoran Desert. She's also a co-author of Bats of Oklahoma. Her current work focuses on bat echolocation, call recordings, and analysis. In 2017, her company, Bat Survey Solutions, sponsored the second International Symposium of Bat Echolocation Research in Tucson. And throughout her career, she has provided over 600 training classes on bat survey techniques for over 3,500 students across the United States, Mexico, Central and South America. Amazing, wonderful. Janet, take it away.
Okay, well, it's nice to be invited to talk to you folks today. And what I thought I would do is to study bats. And it's kind of a niche field, but in recent years, there have been a lot of opportunities for people to do some citizen science work with bats. And so hopefully I will be able to inspire you folks who don't already know a lot about bats to maybe make the foray into the sort of dark secret world of what goes on at night while we're not really paying a lot of attention to other wildlife. So I've got a short presentation for you. It's probably about 20 minutes or so. That's just going to go over some of the unique things that we think about when we are studying bats. So I'm going to share my screen, and hopefully I can do this right. I haven't done Zooms for a while. I'm mostly doing Microsoft Teams, but let me see if this will work. Okay. Did I get permissions to do this as a co-host?
You should have it.
Okay, because I'm clicking on share screen and, oh wait, I think it's working. I apparently need to double click. Okay, so what do we have here? A black screen? Is that what you're seeing? Can you guys still hear me?
Yes.
Okay, great. Can you see me or do you not get to see me anymore? I don't know how this works on a multiple monitor. Okay. Anyway, wait a minute. I think I've got the wrong screen on here. Hang on. Oh, shoot. Now I've lost everything. Oh, I'm sorry. I should be better at this by now. You'd think after a year and a half.
You're not alone.
You to be. I'm not alone. Oh, good. At least you can hear me and I'm not muted, right? Okay, so this should be on the right screen, right? Uh-oh, now I've lost everything. Oh, shoot. Oh, shoot. Excellent. Okay, so what I wanted to talk to you today about is just some of the factors involved in studying bats. And not too many of us often think a lot about bats, mainly because not a lot is known about them. And this is because they're notoriously a really difficult group of animals for us to study. And it's really because they live and operate in a part of the world that we don't often venture into. And when we think about wildlife biology, the animals that we know the most about are the terrestrial wildlife, animals that live and travel on the solid parts of the earth, just like we do. And wildlife that is more difficult to study are those animals that, of course, live in places where humans are not well adapted to travel, like oceans, for one. We can't breathe underwater, nor are we efficient, streamlined swimmers like most of the fish. And wildlife that take to the air for the majority of their lives are just as difficult to study because we can't keep up with them for any distance or for any length of time. And then bats are among the most difficult of all because not only do they live in places that are hard to find, but they're also active at times when they're really hard to see. And they're mostly tiny and they're super fast and they are always just out of our reach. So they present a lot of challenges for us to study. We have to figure out how to keep up with them when they fly. We have to develop ways to see them at night. We have to discover their cryptic roosts, and we need a way to listen to their mostly silent calls. And today, study methods for bats are generally roost surveys, physical capture surveys, and recently we have branched into acoustic surveys where we can use ultrasonic microphones to record and then sophisticated electronics to analyze their echolocation. Ideal for bats that form large conspicuous colonies. Think of things like Carlsbad Caverns. They also work well for species that are easy to identify because they've got some obvious characteristics about them, like large ears or maybe special roosting patterns. The best thing about roost surveys is they're super helpful because they're repeatable. And that's because most bats are very loyal to their roosts, and it allows us to figure out if populations are increasing or if they're decreasing. We can also figure out the gender of the populations and their reproductive status. And then we can see how their choice in roosts change during the season. And we can figure out when they migrate to and from different areas. But sometimes roosts are really difficult to survey. And that's because a lot of bats roost really deep within caves or within mines where people can't safely enter. And a lot of times entering roosts requires specialized training or equipment to be safe underground. And for large populations of bats, counting bats can be really difficult. And it's true for bats that are just stationary, hanging out on a ceiling. And also there are a lot of species of bats that don't even ever enter caves. Some of them have adapted to living in man-made structures, either buildings or bridges. And other species of bats don't like to be near people at all. They prefer to roost in forests. They either hang out in amongst the vegetation of trees, like red bats do, or they roost in cavities and crevices in trees, like endangered Indiana myotis. And then the other big problem with bats, especially species that live in temperate locations, like where we live in the United States and up into Canada, is that they need very different roosts throughout the year. In the wintertime, they need cold but not freezing roosts that have very stable temperatures. And this is because they can let their bodies cool down to that of their surroundings, so they can lower their metabolisms and live off a tiny amount of fat that they store for the entire winter. But then these same species in the summer are seeking out the hottest possible roosts, especially females that are rearing young, where they can find warm air traps that make incubator-like conditions so that the growth and development of their babies that are called pups can occur very rapidly in just a single season. And even if we can find bats in their roosts, they're still difficult to study. And because the main problem is that entering a bat roost is always going to cause a bit of disturbance. And disturbance could lead bats to abandon a site. So our roost surveys have to be really careful. Our well-known hibernation sites are surveyed only once every other year so that we don't risk waking up the bats and forcing them to become inappropriately active and burning through their fat reserves. And maternity roosts are also carefully surveyed, usually just by using summer exit counts instead of entering them. Because if you enter a maternity roost, the flightless young can become scared and fall from the roost and they can't get back up. And because we still don't completely understand migratory behavior for most of our bat species, it's really difficult to survey transitory roosts that bats are using for only a couple weeks out of the year, but are still really important to them. Also, bats that don't congregate in large numbers, like the tree roosting bats, we call these bats over-dispersed because they're not terribly loyal to any one roost, and they'll move from roost to roost throughout the season. They're really difficult to find and even more difficult to count. So for a lot of bats, roost surveys aren't really the best method for understanding where and when we can find them. So we also use capture surveys. And generally bats cannot be captured at roosts, because that's a major disturbance and they might abandon the roost. So we have to try to encounter them where they fly and forage. The big benefit of handling bats is that it allows us to collect really important data about the bats, such as their body size and their body weight. And we can also record their gender and their reproductive condition, as well as their age. And this helps us to learn a lot more about the population dynamics about a species and the habitat associations for different species. But it takes quite a bit of effort and quite a bit of specialized training to be most effective. The main techniques that we use to capture bats involve mist nets and harp traps. And mist nets are very fine nets made out of nylon that are stretched between two poles. The bats fall into the net and are held in a hammock-like pouch until we can walk up and untangle them and identify them and record them and release them. The nice thing about mist nets is that they're really lightweight and they're easy to deploy. But the bad thing about mist nets is that they're very obvious to bats, especially bats that can echolocate. And the ones that are the most sophisticated echolocators are very, very difficult to catch. And also, every single bat that you catch has to be tediously untangled. To catch a bat with a mist net, we have to be really sneaky. It's like being a motorcycle cop with a radar gun hiding in the shade of a school zone. Bats generally have regular travel corridors, just like we do when we drive around town. And when they're flying along their commuting route to their favorite drinking spot or to their feeding area, they tend to get really lazy about echolocating and they fly by memory. And they look out for big things like tree trunks and brick walls, but not the fine mesh of our nets. So we're catching bats that are unaware, just like the motorcycle cop catches us as we're speeding. But it only works the first time. And also, because our nets are essentially ground-based, they're going to work best on low-flying bats, and they'll completely miss species that fly high above the forest canopy. And then harp traps are another way that we use to catch bats. They were especially designed to use in front of cave entrances or other places where a lot of bats can be caught in a short period of time. And the bats are trying to fly through the harp trap, and they get tripped up and fall into a catch bag underneath. And then they hang out and don't need to be tediously untangled. The trap is made up of two different frames about 4 inches apart, and each frame is strung vertically with up to 60 lines of fishing string, and the lines are about 1 inch apart. And the bats are flying through the cave entrance or through any kind of small flyway, and they'll detect the first set of strings, and then they'll twist sideways to pass through. And then they'll open their wings in between the two sets of frames, and they'll crash into the second set of lines, and then they'll fall down into the catch bag. It seems kind of like an improbable way of catching bats, and it only works where bats have to travel through a very small flyway, but it's super effective. Of course, again, the first time you catch a bat. They clearly know the trap is there, but they think they have the skill to fly through because they're not anticipating the second frame of strings. And they'll work in cave entrances, but they'll work outside in the forest along trails in an area where the entire trap can fill the available flyway so that the bats can't fly through any empty spaces that are above or below or beside the trap. But really and truly, bats are generally very difficult to capture, and that's because they will learn capture techniques and they'll remember them sometimes for years. And so mark recapture studies show that it's very difficult to catch the same individual more than once. When we're capturing bats, we're mostly only capturing the naive individuals. So it's hard to encounter the same animal over and over again like you can with banded birds or with tagged rodents. A lot of bat biologists say that everything we know about bats is based on the stupidest bats that we can catch. So luckily we have this other method for catching bats, and that is by making acoustic recordings of their echolocation calls. And then we look at these recordings later to identify what flew by. And the really best thing about acoustics is that the behavior of the bat is unaffected by the deployment, unlike traps or nets or roost surveys that can cause disturbance and cause bats to avoid the area. But the bad thing about acoustics is it's still a really young and evolving science. And the more we use it, the more we learn just what we don't know. And acoustic surveys can tell us just how much bat activity there is throughout the night. And by analyzing the recordings, we can figure out things like relative species diversity and activity levels between two different areas. And we can also talk about different habitat associations and where the greatest diversity is found and the most activity might be for certain species. And that's because anytime a bat is flying, it has to be echolocating. So what we're doing is we're essentially eavesdropping on these bats that are out there anyway. There are three main ways that we use acoustics to survey for bats. One is passive surveys, and with these, we put out our high-frequency microphones on tall poles, and we let them collect data all night, every night for weeks or months or sometimes even a year. And this way we can see how bat activity will change over time. But the problem with passive surveys is we don't know if we're recording the same individual 100 different times or 100 different individuals just once or something between. So we also use mobile transects to get at absolute population numbers. With our mobile transects, we put our microphones on vehicles and we drive through a habitat at a constant speed that's just a little faster than what a bat can fly at. And so every time we get a recording, we can be certain that it's from a unique individual. But the problem with mobile transects is that they can only usually be done along roads, and not all bats like to fly along roads. And then we also use active recording, where we can simultaneously hear or see the echolocation call coming in, and we can observe the bat. And just like bird watching, it takes a lot of practice and skill to become really good at, but by looking at the echolocation call, while we're also seeing the size and wing shape and even the color of the bat, we have a lot more information to make a confident identification. And there are a lot of different types of bat detectors out there that can record and store echolocation calls. And some are better for passive or mobile or active monitoring. And all of them are really pretty sophisticated and they're kind of expensive. And they can take quite a while to learn how to use and how to set up properly. They are more expensive and harder to use than mist nets, but they've become really indispensable tools for bat surveys. And the best thing is that now our newest detectors are microphones that you can plug right into your smartphone or a tablet, and you can watch echolocation calls being translated by special apps for your tablet. And it's going to tell you right then and there what kind of bat is probably flying by. And lucky for us, bats are loud. They are very loud. They use super high frequency sound that we can't hear, but if we could, they would be as loud as a triggered smoke detector. Bat calls are also very discrete. They have a distinct beginning and ending. It's not a continuous roll of sound like, say, a bird call. And they're also very pure in tone. They're either made up of a single frequency or multiple frequencies. They're on a sliding scale. And these are represented by the blue lines in this image. And with digital processing, we can make spectrograms and oscillograms, which is the green waveform below the blue lines in this image. And this shows us how the frequency and intensity of the bat call can change over time. So in this image, we have the frequency in kilohertz along the vertical axis and the time in milliseconds along the horizontal axis. And you can see that this particular gray bat has produced 15 discrete echolocation call pulses that sweep roughly from 80 kilohertz down to 40 kilohertz in just 1.7 seconds. But when we start talking about kilohertz and milliseconds, that's kind of beyond what we humans can really appreciate, because we can hear sounds from about 20 hertz up to 20 kilohertz. And our ears lose their sensitivity when the length of a sound is less than 100 milliseconds. And most of our bat calls are between 2 and 20 milliseconds, and they're almost all above 20 kilohertz. So even when we can transform bat sounds into a real-time sound picture, our senses don't really function with a fine enough resolution to figure out the characteristics that these calls might have, kind of like what you would look at the characteristics of what a bird has, wing bars or eye patches. But lucky for us, the short bat calls are also separated by these long periods of silence. So if you look at these 5 millisecond long bat calls, they've got a period of silence that is about 100 milliseconds long before the next call pulse. And by eliminating the long periods of silence and using the space that they take up to expand or slow down the actual bat call into those silent spaces, we can start to resolve greater detail about how the frequency and the intensity of the call changes over these really tiny time periods. And we call this a compressed view of a spectrogram. And by zooming in on an individual call and its corresponding oscillogram, we can show the detail of the amplitude difference in the waveform. So you can see how the amplitude or the loudness of the call changes over time from the low, quiet blue values to the high, loud red values as the volume of the call becomes greater before fading away. And by looking at pictures of bat calls, we can also learn a lot about how bats use echolocation. This is a sequence of pulses that's shown in compressed view, and it's called a feeding buzz. It's how a bat echolocates when it's catching an insect or approaching a pool of water so it can get a drink. When we look at the same sequence in real time, we see how the buzz got its name. The calls are produced so fast that the silence between the calls becomes almost non-existent, and this actually sounds like a buzz. This display right here shows a 3-second bat call, and the buzz starts at the 1-second mark and lasts for only half a second before the bat picks up a regular echolocation style as it goes off looking for another bug. And bats also produce directives. These are like car horn honks, and they use them to communicate with other bats. They're not necessarily used for echolocation. The bat's not trying to get a usable echo off of the sound. And they'll often make these social calls or directives when they're flying with other bats. And sometimes they can be really species specific and give us a good idea of what species is out there flying. But even though bats are loud, sometimes they're just too far away from our microphones to make good recordings. This is actually a compressed view call, but it doesn't have any of the structure that we're looking for, simply because the best portions of the bat call, the loudest portions, were too far away. It's kind of like the blurry pictures that we take when we're too far away from our subjects. We can't always tell what kind of bat we have or what it's doing when we get a poor recording. And even though we call our recorders bat detectors, they really will detect any high frequency sound. And there's lots of other animals that produce high frequency sounds at night, like crickets and katydids, even coyotes, some night birds, and even rodents will make high frequency sounds. And that's the big problem with acoustic surveys, is that not all recordings we make are useful, because not all echolocation calls that different species make are actually species specific. So we can't use everything that we collect to identify different species of bats. And that's because bats are using echolocation as a tool, not as a unique identifier. And when different species have to solve the same information need using their sounds, they converge on similar call types that are not as species specific. So you can think of echolocation as being a lot like an adjustable beam headlamp or a flashlight. When the beam is really diffused, like on the right-hand side of the photo, it lights up a large area, but you can't see a lot of detail. But then when the beam gets really focused, like on the right-hand image, you can see a lot of detail over a very small area, but you kind of lose your peripheral vision. And bats are doing the same exact thing with their echolocation. They use a long-range, low-frequency call when they're just navigating, but when the bat shifts its focus and tries to get more information about an object of interest, it changes its echolocation call to a more detailed pulse type, a type that's really similar amongst many different species. And that means that bats have really diverse echolocation repertoires, and that not all the calls they make can help us to identify them to species. So the big takeaway is that bats are not birds. They don't always make a call that is recognizable to species. And any bat may emit calls outside its typical known echolocation repertoire, and sometimes can mimic the calls of another species. And as a result, a lot of recordings are going to have ambiguous species classifications. They can't reliably be assigned to a single species with confidence. Because most bats are producing echolocation call pulses every time they take a wing beat, when they're just flying normally, different bats are going to tend to have different echolocation call characteristics, especially when they're not performing any fancy navigation or feeding call type. And we can teach ourselves to identify bats by looking at their echolocation calls while they're commuting, just like you can identify birds by using binoculars when they're either flying in good light or when they're perched and being really still on a branch. It seems really overwhelming at first, but we can learn to identify bat echolocation by putting their spectrograms into different groups according to where the lowest frequencies of their calls fall on a scale. In the United States, there are three main groups of bats. We have those with calls that take up the space from 40 kilohertz and above, and those with calls in between 20 and 40 kilohertz, and those bats that have calls that fall around 20 kilohertz and below. And different species are going to fall into these three different groups. And oftentimes the shapes of their call pulses will help us to identify different members of a single group. For instance, red bats in the 40 kilohertz group produce these really curvy calls that fluctuate randomly in the 40 kilohertz and above range. And canyon bats make curvy calls too, but they stay at a constant frequency of 40 kilohertz or above. And then we have a bunch of species in this group that have pointy tails at the end of their calls, and they look like daggers. These are our many myotis species. Some of them, like the Yuma myotis, have calls that are just above the 40 kilohertz line. And other bats, like the little brown myotis, have calls that dip just below the 40 kilohertz line. And then we have the species that have calls that are mostly in between the 20 and 40 lines. The big brown bat is a species that makes smoothly curved calls in this range. And the silver-haired bat is a bat that makes calls that are bent in the middle, and they look like hockey sticks. So they're not as smoothly curved as the big brown bat. And then big-eared bats make short little calls that line up in between the 20 and 40 kilohertz line, like little tally marks. We also have bats that make calls that sit on the 20 kilohertz line or dip below. One of these is the Mexican free-tailed bat. It's a really common species that lives under bridges and in a lot of caves and in stadiums and in parking garages. And it makes very flat calls right on that 20 kilohertz line. The hoary bat is like a red bat, but bigger, and it makes really curvy calls that often are irregular and bounce around just above and just below that 20 kilohertz line. And our biggest bat in the United States, the mastiff bat, makes calls that are almost always below the 20 kilohertz line. By thinking of bats in these different call frequency groups, you can start to teach yourself how to identify echolocation calls on a spectrogram. And you can collect these on a handheld iPhone or Samsung phone or a tablet, or on some of the bat detectors that also produce simultaneous spectrograms. So you see, soon you can be just as good at recognizing bat echolocation as you are at recognizing birds. We've been able to collect echolocation call recordings from known bat species to make computer algorithms that will automatically identify recordings from wild bats. And these software programs also allow us to display the echolocation calls in real time. And we can generate different analysis outputs from huge batches of recording data. And it helps us to make species ID more of an automated process. So even though bats are super difficult to study, bat surveys are always fun, mainly because you never know what you're going to get. It's kind of like Christmas for me when I open up a file of echolocation call recordings, or when I open up a MistNet to see what kind of bat I have. It's like opening up a present. And that's really the beauty of just about any natural history and wildlife study, is that simply being routinely present and aware in an area, it teaches you a lot about what's going on. And you don't even have to be a professional these days to study bats. We've got several citizen science projects where you can participate and help real scientists and data collectors learn a lot more about bats and their habits on local and even continental levels. One of these is the Backyard Acoustic Survey, the Bat AMP project. It's an online portal for uploading data summaries from any bat detector, including the smartphone detectors, so that it can be aggregated. And this gives us a much better understanding of migratory movements of bats and different seasonal activity of different species. And this is information that's really difficult to collate unless we have a large number of collectors that are across a broad area. All you have to do is put a bat detector out one or more nights at any location across the continent at any time of the year, and then view and summarize and upload the data. Every year, more and more people are participating, and now we have over 200 contributors that have collected over 30 million recordings from almost 250,000 nights with more than 3,500 bat detectors, and they've identified nearly 6.5 million bat passes from 36 different species. And now, for the first time, we can generate continent-wide information about the seasonal and annual occurrence of species, how they change from fall to winter to spring, and how they change from year to year. And we can also get an idea of the total species richness on nightly levels, on seasonal levels, or annual timeframes. And we're super lucky in Arizona because identifying the numbers and species of nectar bats that visit our backyard bird feeders at night is also an important citizen science project. It was first started over 10 years ago in the town of Marana outside of Tucson, and it has expanded throughout the entire range of the nectar bats in Arizona. They go all the way up to the Grand Canyon. It's currently being maintained by the Arizona Game and Fish Department, and you can go to their backyard bats section of their website to download more information about participating in the project and to get data forms. Of course, bats are harder to watch than birds. They're even much faster than hummingbirds. But just like we are at our fast food drive-through windows, the bats will come back again and again. So patient observations are rewarded with lots of views of seldom seen animals. And once they key in on feeders, and we don't really quite know exactly how they find feeders, but I have put feeders up one night and they've been drained in a single night when they haven't been there for weeks and months before. They can become habituated to porch lights and even video cameras and camera flashes. So we can not only get great views of these animals, but we can also determine which species are visiting because they look quite different on film. So it's a really exciting project that you can participate in just by having a hummingbird feeder out in your backyard. And there's lots of other information about bats on the web. I do training classes to teach people different ways to study bats. You can also, if you're interested in putting up a bat house, or if you're interested in getting bats out of your house, you can learn more about bat houses and nuisance bats at batmanagement.com. And then at batcon.org, you can get a whole lot of information about bat natural history and conservation. And then, of course, you can always email me if you've got questions about your local bats, or if you want to learn more about how to participate at a local or regional level on some of the citizen science projects that are out there. So that is what I have for you this evening. And I can take any questions if you want as well.
That was wonderful. So Janet, I do have a question because I've got bird feeders, and I too have noticed that bats do come to visit, but I haven't set up a camera yet. But I'm kind of curious, what smartphone detector do you have a suggestion on what would be a good one to start with?
There is the entry level one, the most affordable one is called the Echo Meter Touch, and it's available from Wildlife Acoustics. And so if you just Google Echo Meter Touch or Wildlife Acoustics, they make it for both the iPhone and for the Samsung. And that is a really nice, just plug and play. It always works. And you can collect data on your device. You can offload data either through AirDrop or through iTunes or over the internet. And then you can look at the data and upload it to the BatAmp project. Or they're really just great interpretive tools too. If you are out at night looking at owls or something, it wouldn't hurt to have a bat detector out too to see what's going on.
I will order one soon.
Okay.
I have a question too. So for the harp traps, I understand how they work in terms of the actual stopping their mobility, but once they fall into the bag, what is keeping them from flying out of the bag? Like, how does that functionality work?
Oh, it's really clever. The bag is a really nice heavy duty canvas, and on the edges, the top edges of the bag, they sew a little piece of plastic. And what the bats do is they can't really cling to the plastic, but they crawl up the canvas and then they roost near the top edge underneath the plastic. And they're just going to hang out with each other and chat to each other and say, oh, can you believe what happened to me? And then they just wait for you to pluck them out.
Awesome, thank you.
Mhm.
And in terms of the was it spectrograms? Is that what you called them? Or
Right.
did it take you to really get a feel for identifying the bats? Like, what is the learning curve for doing something like that and saying, oh, that's a hoary bat?
Yeah, I like to kind of equate it to bird watching. You know, at first you didn't know if you were looking at a woodpecker or a dove or a raven, right? And then you kind of notice, oh, well, the ravens are really dark and the woodpeckers have this strange flight style and the doves are more direct. And, you know, and then you can move on to things like, you know, your finches and your hummingbirds and your vireos, the things that are where you have to use your powers of observation at really minute levels to figure out what's going on. So, you know, there's definitely a learning curve. But now I'm sure, like, if you've done birds for a long time, just out of the corner of your eye, you know if you've got a little verdin flying by.
Right. Okay. Thank you. Also, this is amazing, and I'm obsessed with you and this project. And Juanita wanted me to relay her hello because she said she's actually met you in person as well, but she couldn't be here tonight.
Who's that?
Juanita Armstrong. She said that I think she met you.
Okay, yeah.
Okay, yeah. But all right, thank you.
Sure.
I have a question. So I've been looking at like phenomena around phenology, so like date of first flowering and things like that.
Mhm.
And I'm wondering, like, and then anyway, we were talking just about how like that impacts then, the larger system of like possibly pollination and so on and so forth and other life events of other species. So I'm wondering if you're doing any research around that and if you've noticed anything just with bats related to that.
Yeah, that's actually where I got my start with bat research. Was I worked in Mexico on the Sonoran coast, where we've got the cardon, the saguaro, and the organ pipe, and they all bloom around the same time of the year. They all have nearly the same size of flowers and the same amount of nectar production, but they have different blooming times. Where the saguaro would they would bloom after it was full dark, and they would stay open until like noon or so. And so they were kind of hedging their bets for both nocturnal pollinators and diurnal pollinators, whereas the cardon were much more nocturnal pollinator related. And it wasn't just bats, but large moths would visit as well. And, I mean, this was back in the old days of bat research. This was in the 80s, but people are continuing on this research. And as you probably notice, even, just out of the corner of your eye each year, there are some years where we have tons of saguaro blooms and other years where it seems like we hardly have any. And people have been continuing this on. They're using much more sophisticated techniques rather than stomping through the desert with ladders and legal pads. They're flying drones over these big columnar cacti fields and counting how many flowers bloom each night. And I mean, it's just truly amazing what's going on. But what I think happens with these bats and these habitat associations is that these populations are so mobile, especially with these migratory nectar bats, that things fluctuate on a multi-generational timescale. I've been studying and catching bats in the Chiricahuas for 30 years straight now, every May and June. And we see wild fluctuations in the numbers of bats that we get from year to year. And for the longest time, we thought we were actually seeing a major decline in our canyon bat populations because we were getting fewer and fewer every year. But if you look at it in like little 5-year increments, we're seeing a huge increase in canyon bat population based on our capture surveys in just the past 5 years. And so I think it's really a hard question to answer because there are so many reasons why you may or may not have bats in this area or not that area from year to year. And that's why these long-term studies like this Bat AMP project are so good, where we can passively listen to these bats as opposed to like looking at their roosts or looking at their capture records. Our passive detectors are not going to be influencing the bats at all. But there again, it's a very young science, and we've only been doing this for like 10 years, which is just a fraction of the time that we've been catching bats out in the landscape. So yeah, there's a lot more questions than we have answers for right now, because there's almost 50 species of bats just in the United States and Canada. And there's about 200 or 300 bat scientists that are actively researching. And then of course, a bunch of graduate students and whatnot. There are over 1,200 species of bats worldwide, and there are fewer than 1,000 bat researchers. So there's a lot of stuff that's not getting looked at with these animals.
I had a quick question. I'm kind of new to bats, and I was just curious what the bat's lifespan was.
It depends. Some of our bats will live 10, 12, 15 years. Our longest lived bats were banded as adults and then recaptured at hibernacula at caves during cave surveys in the winter, and have shown to be 38 and 37 years old. And this may have to do with the fact that they hibernate, and for 6 months out of the year, their heart rate goes from 200 beats a minute down to about 6 beats per minute. So they're not really wearing themselves out much when they're hibernating.
Okay, I'm gonna ask another question since no one else is. So obviously Arizona population is going crazy and urbanization is really on the rise. Since you've been tracking this the last 15 years, have you noticed a large flux in population related to urbanization or even location of the bats?
One thing that I have noticed just in my own backyard, because I do winter-long backyard monitoring, and I've lived here for 20 years now. And it's been about, I would say maybe 10 years that it's been super organized, because like I said, this is still kind of a young evolving science, this recording echolocation calls. And I am seeing a large increase in certain species of bats, like Western yellow bats. These are bats that roost in the dried palm skirts, the fronds that collect along the trunk of the trees. And more people are using these as like ornamental plants, landscape plants. They're also being protected in a lot of the parks that we have in towns. And there's been a big push amongst landscapers to not trim these trees during the summertime when the bats are rearing young, and only trimming them in the wintertime. But we've seen a large increase in yellow bat recordings in suburban and urban areas, both in Phoenix and in Tucson, and they seem to be mostly present year round. I was getting them up until the end of November last year, just in my backyard here. And our nearest palm trees are probably about, I would say 100 meters away from my property. But these animals are pretty mobile. We also are seeing good increases of Mexican free-tail bats in some of the area bridges and parking garages, stadiums. There's a couple of roosts in the Phoenix area. I think one associated with like a dam near the Salt River project. Yeah, and a lot of these large roosts are being protected, if not formally, then at least informally with information. Just educational information. And people are really starting to appreciate bats a lot more. And rather than taking a swing at them with a tennis racket when they fly into their house, or, squirting them with the hose when they're roosting on their front porch, they're kind of living and let live sort of thing, or putting up a bat house to maybe get them off the porch and into some place where they're not pooping on the welcome mat. So it's been kind of nice. And the lesser long-nosed bat, one of our nectar bats that comes to the feeders, has just been taken off the endangered species list. It has been listed as endangered since the early 80s, I believe. And in 2016, 2018, it was completely, it wasn't delisted, it was completely unlisted, simply because we've learned a lot more about its migratory habits, and that it's not always present in certain caves that have been monitored when we thought, oh my gosh, the populations are declining. So that's a good success story too.
What would you say is your favorite part of the bat work that you do? Like, what is, when you bring to mind, like, oh, that was my favorite moment, what were you doing in that moment?
I just love being outdoors at night. I mean, it is just so much stuff goes on at night, and so few of us have a good reason to be out at night.
Right.
I mean, really, I mean, the weather is beautiful here in Arizona almost year round, so you could be out at night. But, when you're sitting out at a mist net waiting for a bat to hit it, it's kind of like fishing. It's a lot of minutes of boredom punctuated by seconds of intense excitement. But, just being able to watch the stars and shooting stars and constellations and seeing the Milky Way, and then all the other creatures that are out at night, that we just aren't aware of. I did some nighttime monitoring in my backyard, and I had no idea how many foxes we had out here. I always think of that as being like a little forest animal, but they're out here in the desert here. And yeah, I like being out at night.
Thank you.
Do we have any risk of white nose disease here in Arizona, our bats?
We have had some confirmed cases of white nose in a couple species of Arizona bats. I know one was a myotis up near the Grand Canyon. Now, this is a fungal disease. It's a novel fungal disease that was first noticed in 2006 outside of Albany, New York. And it has spread just geometrically since then. And it was identified about 2 years later, and we found out that it's a cold-loving fungus where it replicates ideally at temperatures around 10 degrees C, which I think is around 40 degrees Fahrenheit. And that happens to be the temperature at which most of our hibernating bats will spend the winter. And when they hibernate, they're all packed really densely together in these cave ceilings. And we had hibernacula in the eastern United States that had 300,000, 500,000, 1.5 million bats hibernating. And it was just like a petri dish of media for this fungus. And so it replicated out of control, and it killed a lot of bats because it woke them up, and they ended up burning up all their fat reserves. And then they couldn't last until spring, and they starved to death in the wintertime. So we lost between 5 and 6 million bats in the eastern United States. And it has progressed southward into the Atlantic and Gulf Coast states. It has progressed westward into the Southwest, into Texas, Oklahoma, Arizona, New Mexico, and into the Northwest, into Washington and Oregon and Northern California. We haven't seen the huge die-offs of bats here in the Western United States, mainly because we don't have these huge hibernacula out here. We've got the largest known hibernating roost of bats is a bunch of cave myotis in the Huachuca Mountains, and there's 200 bats there. And, you know, they go into hibernation maybe at the end of November, they come out maybe at the beginning of March. So they have a very short hibernation time. We've got bats, we had bats in the Eastern United States that would go into hibernation in late September, and they wouldn't come out until, you know, mid-May. So the length of hibernation is much longer out there, and the dense packs of hibernating bats was just perfect for the fungus to grow and develop and replicate and really cause a big problem. And I think here the bats that are getting it, they may have the fungus, they may be disturbed by it, they may be inappropriately active, but they have other options for finding food in the wintertime during warm snaps. A lot of our bats also hibernate. They'll migrate elevationally in the West. So they'll spend the summer at low elevations around 2,000, 3,000 feet, and then they'll go up to 6,000, 7,000 feet to hibernate. And that may be over a very short travel distance. So if you've got bats on Mount Lemmon, if you've got bats in the White Mountains, if you've got bats up in San Francisco peaks that are hibernating, they can come down to the basin and feed in the wintertime on a warm day and replenish that fat reserve. So it may not be as big a problem to the bats out here, but it could also be that the bats, because they're hibernating in such small, out of the way places, if there are major die-offs, we won't notice it. So there again, that's why this acoustic monitoring is so important when we do this year-round monitoring, and we can see how activity levels and numbers of echolocation calls that we're picking up for different species throughout the winter, if it radically changes from thousands to tens or single digits, then we're going to know something is up. But yeah, it's a tough question. We're not quite sure what's going on with our Western bats. Fingers crossed, it's not going to be a big problem out here.
Any other questions for Janet? Janet, anything else you want us to know?
Oh my gosh, I mean, you know, I could talk for a week. There's so much to know. But
Well, if there's
maybe
no
I'll
other
see you
questions.
next-
Oh, go
I'll
ahead,
see you
Janet, I'm
next
sorry.
year. Maybe I'll see you next year and we'll have more things to say.
Absolutely. Well, thank you so much for being with us tonight. I know I thoroughly enjoyed it, and I'm sure everyone else did as well. And I think you just recruited some new bat lovers.
Okay, well, I hope the recording worked, and I'm sorry about my technical difficulties. I'm so looking forward to getting back in person.